Okay, hello to all of you. Let's discuss chapter number 10. And again, as I cannot make very big videos, I'm going to go over some of the important points of this chapter and other things you can go over. And if you have any question, you can let me know. So again, this chapter is about the metabolism, and you have already done all these things. So these things are little in more detail. So basically, in the last lecture, we talked about in this chapter, we are going to talk about the further stages in the aerobic respiration. Glycolysis is the first stage, which is common in both the aerobic as well as the aerobic respiration or the cellular respiration as well as the glycolysis. So next step is when it gets converted into acetyl coenzyme A. So let's go over and talk about that process. So basically, you can see here, we know that glucose converted into pyruvate. This takes place, this is glycolysis, it takes place in the cytoplasm, and then the pyruvate is converted in the mitochondrial matrix. In the mitochondrial matrix, pyruvate is converted into acetyl coenzyme A, and then this acetyl coenzyme A goes through Krebs cycle, in which 8 reactions are involved. We are going to discuss that. And so in the Krebs cycle, actually, the food is almost completely utilized, and at every step there are involved some of the electron carriers, NAD plus and FAD. They are the ones which are carrying these high energy electrons, and these high energy electrons, they are taking them to the inner membrane of mitochondria. And so what's so special about the inner of mitochondria? It's the electron transport chain which is present here, and also this special enzyme called ATP synthase, where the ATP is produced. So this is overall cellular respiration. You already know that. So these are the 5 steps of your cellular respiration: glycolytic pathway. Then pyruvate is converted into acetyl coenzyme A. Then acetyl coenzyme A enters into citric acid cycle, and then we know that there is electron transport chain which is there, which is which involves basically the proteins which are embedded in the inner membrane of mitochondria, and the flow of the electrons creates the electron electron chemical proton. It basically creates the proton gradient, and as a result of that, when these protons they fall back through ATP synthase, there is a production of ATP, and this process is called oxidative phosphorylation. So let's go and talk about a little bit the structure of the mitochondria. As you already know, mitochondria is the powerhouse. So this is the basic structure of mitochondria. There is the outer membrane, then there is the inner membrane, and in between outer and inner membrane, this space is called intermembrane space. This is all called matrix region. In this matrix region is the DNA, the ribosomes, and our conversion step, pyruvate to acetyl coenzyme A, Krebs cycle, in matrix. And this invaginated inner membrane, is called cristae, and this space is called intracrystal space. So this is the basic structure of mitochondria. The outer membrane, it has porins. So we have already discussed in previous chapters what porins are, and intermembrane space between inner and the outer membrane. Okay, this is all what I just said. And the ATP synthase which is present, it has two subunits. It has FO and F1. So these FO, F1, uh, these are the two subunits which make the ATP synthase. And FO is actually embedded in the membrane, and F1 is actually protruding out in the matrix. So this is the ATP synthase. Okay, so it's made of two subunits, FO and F1, and we're going to talk about them in detail. So F1 complex is attached by a short protein stalk to a FO complex. So FO and F1 complex is an ATP synthase that is responsible for the most ATP generation in the mitochondria. And let's go and talk about the citric acid cycle. So citric acid cycle involves 8 reactions. Let's go over them. Again, you need to know the enzymes which are involved at every step. So basically, you know that acetyl coenzyme A combines with oxaloacetate. Oxaloacetate has carbons. Acetyl coenzyme A has 2 carbons. So these two compounds, they combine and they form citrate. So citrate has 6 carbons in it. Okay, and the enzyme involved is citrate synthase. Then citrate gets converted into isocitrate, and the enzyme involved is oconitase. Then isocitrate, it gets further oxidized. Okay, isocitrate has 6 carbons. It gets oxidized, which means it loses electrons, and those electrons are taken by NAD plus, and NAD plus is converted into NADH. And there is also one loss of there's a loss of one molecule of carbon dioxide. So which means that isocitrate has 6 carbons. When it loses one carbon, now it is getting converted into a compound which has 5 carbons. So isocitrate gets converted into alpha ketoglutarate. Alpha ketoglutarate has 5 carbons in it. And this reaction is catalyzed by enzyme isocitrate dehydrogenase. Then alpha ketoglutarate is converted into succinyl coenzyme A. Here again there is a loss of another molecule of carbon dioxide from a compound which is made of 5 carbons. It's converted into succinyl coenzyme A, which has 4 carbons. And again oxidation is taking place here also at this step. And this reaction is catalyzed by this enzyme called alpha ketoglutarate dehydrogenase. Now succinyl coenzyme A is then converted into succinate. And this is the first reaction where the first molecule of ATP is produced. And this molecule of ATP which is produced, it's produced by substrate level phosphorylation. And then this succinate is converted into fumarate. And here again oxidation is taking place. Succinate loses electrons and gets converted into fumarate. Here the electron carrier which is involved is FAD, and FAD gets converted into FADH2. And the enzyme involved is succinate dehydrogenase. Then fumarate is converted into malate, and the enzyme involved is fumarate hydratase, and then malate is converted back into oxaloacetate, and here again oxidation takes place. So malate is oxidized and it gets converted into oxaloacetate, and it's catalyzed by the enzyme malate dehydrogenase. So this is basically a Krebs cycle, and there are 8 reactions. You need to know what is happening at every reaction, and you also need to know the enzyme involved. So this is all what is written here, what I just discussed. So now we are going to talk about all these electron carriers. They are taking these electrons to the inner membrane of mitochondria, and in the inner membrane of mitochondria is embedded the electron transport chain. And we are going to talk about these proteins. They actually are present in the form of some complexes. So let's talk about the different kinds of proteins which are present in the electron transport chain. So they involve flavoproteins, iron sulfur proteins, cytochromes, copper containing cytochromes, and coenzyme Q. So some of the features of electron carriers: all proteins except coenzyme Q are proteins with prosthetic groups which are capable of being reversibly oxidized and reduced, and they are all hydrophobic. So something very important to know about these electron carriers is, which is basically all the proteins except coenzyme Q, that they have prosthetic groups in them which can get oxidized and reduced. So basically, as you know that these electron carriers, NAD plus and FAD, these electron carriers, they are bringing the electrons, and they are bringing electrons, and they are delivering these electrons to the electron transport chain. And this electron transport chain is made of these kind of proteins, and these proteins have prosthetic groups in them, and these prosthetic groups, they can get oxidized and they can get reduced. So that is why when they accept the electrons from these electron carriers, they get reduced, and then they pass these electrons to the next electron carrier. So when they pass the electron to the next electron carrier, they get oxidized again. So this is the way these electron carriers, they keep on passing these electrons, and the final electron acceptor is oxygen. So eventually these electrons are accepted by the oxygen, and oxygen is reduced to water. So proteins, so these are membrane bound, the membrane bound flavor proteins. They use either FAD or FMN as the prosthetic groups. For example of flavor proteins is NADH dehydrogenase and succinate dehydrogenase. So important character of flavor proteins is it can transfer both electrons and protons. Then we have iron sulfur proteins. They are also called non-heme iron proteins, and they have iron sulfur as the center. So the prosthetic group is iron and sulfur. And the iron atoms in the center of the proteins are actual electrons, and they alternate between Fe plus 2 and Fe plus 3 states. So when they are in plus 2 state, they are reduced, and when they are in plus 3 state, they are oxidized. So they can transfer one electron at a time and no protons. Cytochromes. So they also contain iron, but that iron is a part of a porphyrin prosthetic group, which is a heme. Okay, and there are 5 types of cytochromes: B, C, C1, A, and A3. The iron atom of the heme group serves as the electron carrier and transfers one electron at a time, and they also do not transfer the protons. So this is the prosthetic. This is the porphyrin ring of the heme. So this is the structure of heme, and you can see here is iron. Then we have next category of cytochromes. So they are copper containing cytochromes, and these cytochromes include A and A3. They contain a single copper atom bound to their heme group. So in addition to iron, these cytochromes also have copper. So it associates with iron atom to form a bimetallic iron copper center. So these copper compounds, they can also be oxidized, copper plus 2 to reduce from copper plus 1 by accepting or donating the electrons. So this is some information about these proteins which make the electron transport chain. Okay. The last one is coenzyme Q. This is not a protein. This is the only non-protein component of electron transport chain is coenzyme Q. Because of its ubiquitous occurrence in nature, it is also called ubiquinone. And coenzyme Q or ubiquinone, is reduced in 2 successive steps. First of all, it accepts 1 electron and 1 proton, and it gets converted into semiquinone. And then it accepts another electron and proton, and it gets converted into dihydroquinone. So you can see that this is ubiquinone. So it can in the first step it accepts 1 proton and 1 electron, and it gets converted into semiquinone. Okay. So 1 electron 1 proton makes 1 hydrogen atom, so that is attached here, and in the next step it again accepts 1 proton and 1 electron, and then another hydrogen atom is attached here, and that results in the formation of dihydroquinone. So all the proteins we have discussed, just other than the ubiquinone, all are proteins protein complexes which are present in the electron transport chain, and there are four protein complexes, or we call them four large respiratory complexes, which make the electron transport chain. So this is the first complex, and this is the second complex, third, and the fourth. And all the all the proteins which we just discussed, they are part of these protein complexes. So the complex one is called NADH coenzyme Q oxidoreductase, and it includes the proteins with iron sulfur centers and the flavoproteins which has this FMN as a prosthetic group. This is complex number 2, and this has FAD, so it has flavoproteins which has FAD as the prosthetic group, and it also has iron sulfur proteins. And then complex number 2 is called succinate coenzyme Q oxidoreductase. And then this is the complex 3, which has cytochromes and also the iron sulfur proteins. And this is complex number 4, which has cytochromes and also the cytochromes with the copper in them. One second. Actually, the cytochrome A and A3 has a prosthetic group, which is actually, so the cytochromes A and A3 have the prosthetic group, which is iron and copper. And at the end of this complex 4 is oxygen. So these are the 4 complexes. So now let's see. So basically, NAD plus, it is bringing electrons and it is going to deliver the electrons to the first complex, and then the electrons which are being brought by FADH2, or FADH2 is a reduced form of FAD, they are basically delivered to complex number 2, and after that they flow through complex number 3 and complex number 4 and eventually reach the oxygen. So this is the way how electrons, they flow through these protein complexes, and when these electrons, as you know, when these electrons, they flow through these proteins, there is a pumping of protons from the mitochondrial matrix to the intermembrane space, basically. So these electrons, they will be flowing through these protein complexes, and their energy will be used for the active transport or pumping of the protons from this side to this side, and as a result of that, proton gradient will be formed, and then they flow back through this special enzyme or a special protein called ATP synthase. And we are going to talk about how actually the production of ATP takes place by the process of chemiosis. So let's see. So NAD plus, it is delivering 2 electrons to the complex number 1. So remember, they're bringing 2 electrons. Every NADH is having 2 electrons, and it is delivering electrons to complex number 1. Okay, complex number 1 receives NADH. This complex number 1 is receiving 2 electrons and passing Q. So this is to coenzyme Q via this protein, because this is complex which is made of flavoproteins and iron sulfur proteins, and so it is passing these electrons to coenzyme Q through FMN and iron and sulfur protein, and in this process 4 protons in this region. Okay, so these electrons, you can see how they are flowing. They're going from here and they are actually reaching coenzyme Q, and from coenzyme Q they are going to go to complex number 3, because NADH brings the electrons to complex number 1, then they go to complex number 3 and complex number 4 and eventually reach to oxygen. The electrons which are brought by FADH2, they are going to be delivered to complex number 2. So in the complex number 3, complex number 3 passes electrons from coenzyme, the reduced ubiquinone, to so this is coenzyme Q or ubiquinone. Say it's getting electrons and it is getting reduced. Now this reduced ubiquinone is going to pass the electrons to it passes the electrons from coenzyme Q to cytochrome C. This is cytochrome C, and it passes these electrons to cytochrome C through cytochrome B and cytochrome C1. And then these electrons, and in this process, there are so there are actually 4 in total protons which are pumped from this side to this side. So up to here 8 protons have been already pumped from this side to this side. And then this is complex number 4. It receives electrons from cytochrome C. This is cytochrome C, and via cytochrome A and A3. So this is A and A3, passes them to molecular oxygen. So this is molecular oxygen, which is reduced to water. As more, 2 more, and also here at this step 2 more protons they pump from this side to this side. So in total in this region there are 10 protons, and these 10 protons, because there's a higher concentration of protons on this side because of the pumping of protons on this side, there are less protons on this side, there is a formation of the proton gradient, and these protons they're going to flow through this ATP synthase and they're going to produce ATP. And now we are going to talk about the structure of ATP synthase and how exactly it produces ATP. So that's what I'm going to focus on now. So you can go over and talk, just go over and read about all these. So I'm going to go over, so this is basically shows again the same thing as I just discussed. Now I'm going to talk about the structure of ATP synthase. It has 2 subunits, F1 and O. FO is actually embedded in the membrane and F1 is actually protruding out. So let's go and talk about the parts of this, both FO and F1 subunit of ATP synthase. So I have just made it a little big so that you can see properly. Okay. So this is FO, this is F1, okay. Now FO, the static component of FO consists of 1 A and 2 sub B and 2 B subunits. So this is A and these are 2 B subunits. They are like stalks, okay. The A subunit forms the proton channel and is immobilized in the membrane. Okay, so the protons they pass here, okay. The protons which have been collected, we just saw how the protons are collected on the in the intermembrane space. So they are going to flow through this subunit, A subunit. The A subunit forms the proton channel and is immobilized in the membrane. The B subunit forms the peripheral stalk and are attached both to the A subunit and to the F1. So these are the 2 B subunits and they are attached to this delta subunit of F1. Okay, so this delta subunit belongs to F1. So through these 2 B subunits, F0 and F1 subunits, they are connected. And the mobile component of F1, it consists of a ring of 10 C subunits. So these are C subunits and there are 10, okay. And only one C subunit can form an ionic bond with A subunit at a time. So this is the C subunit and this is the A subunit. So there are some of the amino acids in C subunit, they're negatively charged, and there's some amino acids which are positively charged in the A subunit. And as a result of that, there is a formation of an ionic bond between C subunit and the A subunit. But the C, this ring has actually 10 C subunits. So at a time, only one C subunit forms a bond, ionic bond with the A subunit. Now talking about the F1 part, this part which is, so F no, FO is embedded in the membrane, F1 is actually protruding out. The F1 static component consists of delta subunit, this is the one, plus a catalytic ring, this is the ring. Okay, this is responsible for the enzymatic activity, and it has 3 alpha and 3 beta subunits. So the static part of F1 consists of delta subunit plus catalytic ring formed by hexagonal of alternating alpha and beta subunits. So alpha 3 beta 3 ring is the site of ATP synthesis and is immobilized by the delta subunit. So it's sitting on this. And this delta is connected to B1 of FO subunit. Now the mobile component consists of epsilon, this one, and gamma subunits. So you will see that when this ring will move, this gamma will also move, and it is going to activate this. This is the catalytic subunit of this enzyme ATP synthase, and you will see that when this ring will move, this gamma will move, and this is the catalytic ring, and the beta subunits of this ring, they will be changing the conformation, and as a result of that ATP will be produced. So that's what we are going to talk about. So F1 mobile component consists of epsilon, this subunit, and the gamma subunit, which is this subunit, which form the center stalk that is firmly attached to the C10 ring of the FO. Okay, so now we are going to talk about the beta subunits which are present in this catalytic knob, how they go through different conformations, and how they change into different conformations, and they change into different conformations when this gamma subunit, which is connected to this subunit, when this gamma moves, then there is a change in the conformation of these beta subunits, and as a result of ATP is produced. So there was a scientist called Paul Boyer. He was the one who proposed this binding change model, and he proposed that each of the 3 beta subunits of F1 complex progresses through 3 different conformations. So let us see what those 3 conformations are. The 3 conformations are L, T, and O. L is for loose. So this is the conformation of the beta subunit. This is the loose conformation in which ATP, ADP, and phosphate binds loosely. T is tight for tight. It binds ADP and phosphate tightly, and it catalyzes the formation of ATP. O is open with little affinity for either the substrates or the products. So basically, I think I didn't tell you something here. So basically what happens is, how does this ring moves? As I said, like this subunit has amino acids which are negatively charged like aspartic acid, and this A subunit has amino acids which are positive charged like arginine. So they in this ring there are 10 C subunits. One of the subunit is always forming a bond with this A subunit. When the proton flows through this A subunit, which acts like a proton channel, so where it neutralizes, because proton has a positive charge, it neutralizes the charge. It neutralizes the charge which the amino acid which is there in the C subunit, which is there. It neutralizes the charge on the aspartate which is there in the C subunit. So because of the neutralization of the charge, there is actually a breaking of bond between the aspartic acid and the arginine which is present in this subunit. It breaks the bond, and as a result of that, this ring moves. So when one subunit moves, which means this ring moves by 1/10 turn. So this is the way how this ring moves by the flow of the protons. When this ring moves, this gamma also moves. And when this gamma moves, so this gamma moves, it moves completely, which means it can move at an angle of 360. So whenever this gamma subunit moves, whenever it rotates by 120 degrees Celsius, there is a change in the conformation of the beta subunit. So let's go back to the different conformations of the beta subunit and how it produces ATP. We discussed these are the three different conformations. In the B conformation, beta subunit binds ADP and phosphate loosely. In the tight conformation, it binds ADP and phosphate tightly, and it results in the formation of ATP. And O is with little affinity for either substrate or the products. So substrates are ADP and phosphate, and products are ATP. So Boyer proposed that at any time each of the active site is in a different, and that the hexagonal ring of alpha and beta subunit rotates relative to the center stalk containing the gamma subunit. As I just showed you, this guy moves and it makes this catalytic knob move, and as a result of that, ATP is produced, and I'm going to talk about that right now. And the rotation of the catalytic knob, which contains alpha and beta subunits, is because of the movement of gamma subunit, and gamma subunit is moving because of the movement of the C subunit, and which is because of the flow of protons through FO, the C subunit of the FO. So these are the conformations of the beta subunit. So you can see here, it can go from O form to E form to F form. So the alpha form, ADP and phosphate, they bind loosely. In the O form, they have a little affinity for both the substrate or the product, and in the T affinity, it has tight bonding for ADP and phosphate, and it results in the formation of ATP. So each beta subunit passes through O, L and T conformations as the gamma subunit rotates 360 degrees Celsius. So this I have already discussed. So beta 1 subunit starts in O conformation. So in the O conformation, it has little affinity for both the substrates and the products, which means it has a little affinity for ADP and phosphate, and little less affinity for ATP. So the beta unit starts with O conformation, and when the gamma subunit rotates another 120 degrees, the beta 1 shifts to T conformation. And in the conformation, it has high affinity for ADP and phosphate, and that results in the formation of ATP. And then the gamma subunit rotates another 120 degrees Celsius, returning beta 1 to return to O conformation. So basically, as the gamma subunit is going to move through 360 degrees Celsius, this beta subunits, they will be going through all the three different conformations, and that is and when the beta subunit is in the T conformation, it is going to produce ATP. So this is overall basically how ATP is produced. Okay, so basically we have seen that NADH, when it delivers 2 electrons to the electron transport chain, there is pumping of 10 protons across the membrane. The protons go from matrix to intermembrane space. And for FADH2, it actually, when the electrons, when FADH2 is bringing 2 electrons, and those 2 electrons, they are being delivered to the second complex, there's a pumping of around 6 protons. So assuming that 3 protons must return through the FO and F1 per ATP generated, so which means that when 3 protons they flow through, when 3 protons flow through ATP synthase, there is a production of 1 molecule of ATP. So when around 10 protons are being pumped by the passing of electrons by NADH through the electron transport chain, when 10 protons are pumped, so around 3 molecules of ATP will be produced, because 3 protons, they can produce 1 ATP. So when the 10 protons are there, they are going to produce around 3 molecules of ATP, and there are around 2 molecules of ATP produced when the electrons, when these sorry, so 3 protons, when they pass through the ATP synthase, it produces 1 molecule of ATP. So when 10 protons are going to be passing through ATP synthase, 3 molecules of ATP will be produced, and similarly for FADH2, it results in the pumping of around 6 protons, so which means 2 molecules of ATP. Now this is the overall. So basically in this process, around 36 to 38 molecules of ATP are produced overall. I think that's all what we need to know in the cellular respiration. And if you have any question, you can please let me know.